During my project I've worked across a lot of different departments and different labs, but working at the NEOF lab has been my favourite place to work.
What I liked best about NEOF was being surrounded by other people doing other exciting research on species. It just felt like a nice place to be.
My name's Deborah Dawson and I'm the manager here at the NERC Environmental Omics Facility at the University of Sheffield. Our facility supports PhD students and other researchers who want to use molecular techniques to support their studies of the environment.
The NEOF Visitor Facility has all the equipment to support a wide range of molecular technique. These include DNA sequencing from both individuals and the environment, DNA genotyping to address questions in behavioural ecology and population genetics, metabarcoding for studying diet and biodiversity, qPCR to detect rare species from environmental samples, and our cleanroom facilities for obtaining DNA from challenging materials such as museum specimens.
I'm Gavin Horsburgh. I'm the Wet Lab Manager for the NEOF facility here in Sheffield.
My name's Tom. I'm one of the research assistants that works in the lab. If you'd like to follow me, we'll have a look at some of the equipment.
This is one of our next generation DNA sequencing. We use it to investigate diet using DNA extracted from fecal samples and for environmental DNA projects where we want to discover what species are in a sample of water or soil.
This piece of equipment is used for genotyping. A typical study would involve DNA profiling lots of individuals within a population. We can then go on to use this data to answer questions on their genetics, their behaviour and their evolution.
This is our QuantStudio 12K Flex qPCR machine. In the genomics facility, we use qPCR not only as a means to detect specific species, but also for the quantification of their abundance. qPCR is also a crucial method in the workup for next generation sequencing. Within our facility, we have two purpose-built clean rooms. Visitors working on low-yield samples can extract their DNA and prepare PCR reactions in our clean rooms with a vastly reduced risk of environmental contamination. This is especially important for environmental DNA projects and museum samples.
Our training team run courses throughout the year, both online and in person. Most of these courses cover different aspects of how to analyse omics data, but we also run some wet lab courses. For example, our new eDNA course involves taking water samples from a local pond, bringing it back to the lab, extracting the DNA, and then sequencing and analysing the DNA to see which species are present within the pond. Our team provide one-to-one support for all of our facility visitors, helping them to design their project and to analyse their data at the end of their lab work.
It's simple to apply to the facility. We can help you with your project design, and if you get access to the facility, you can stay for up to 6 months. If you think that these opportunities could suit your research, then please do get in touch with us.